In 1667, a Paris physician named Jean-Baptiste Denis opened a vein in the arm of a 15-year-old boy and let the blood of a lamb run into it. The boy lived. Denis tried it again on a butcher, who also lived, then on a man named Antoine Murat, who shook violently, passed black urine, and died after the third attempt. Denis was tried for murder. Transfusion was banned in France, then in England, and the ban held for roughly a century and a half. Nobody could say why the same procedure rescued one patient and destroyed the next. When transfusion returned, the pattern did not improve. James Blundell, an obstetrician at Guy's Hospital in London, began using human blood instead of animal blood in 1818, mostly on women bleeding to death after childbirth. Of the 10 transfusions he documented over the following decade, about half of the patients died. He used human donors. He worked fast. He understood that the blood had to be warm and moving. None of it mattered. The blood itself was the variable, and nothing about it could be seen. Under the microscopes of the time, the red cells of a donor who saved a life and the red cells of a donor who ended one looked identical. The thing that made the difference was a sugar. In the year 1900, a young assistant at the University of Vienna, named Karl Landsteiner, mixed the serum of his colleagues with each other's red cells and noticed, in a footnote to a paper about something else, that some combinations clumped. The next year he described 3 groups. In 1902, his students Alfred von de Castello and Adriano Sturli found the 4th. Landsteiner had found out why transfusion killed people, and he collected a Nobel Prize for it in 1930. Solving transfusion opened a stranger problem, and it is the one this video is about. A blood type does nothing visible. It does not change your strength, your height, your face, or your life expectancy in any way you would notice. And yet the human species carries 4 of them, in stable proportions on every continent, and the difference between your blood and the blood of the person next to you is older than our species, older than our genus, and older than the split between the human line and the chimpanzee line. Something has been keeping that difference alive for more than 20 million years. Something with a reason. If you're enjoying this story so far, don't forget to hit the like button and subscribe. Drop a comment telling me where in the world you're watching from. I love hearing from you. Most people assume a blood type is a medical category, a label invented in a laboratory so that hospitals could match donors to patients, roughly the way shoe sizes were invented so that shoes would fit. That assumption is half right. The categories were named in Vienna, and outside a hospital your letter has almost no practical consequence. You can go your entire life without knowing it. But the letters were not invented. They were found. The molecules Landsteiner stumbled onto in 1900 had been sitting on human red cells for the entire existence of humanity, and on the red cells of our ancestors long before that. Where the assumption breaks down is geography. If blood types were arbitrary noise, they would be scattered randomly across the planet. They are not. Type B reaches roughly 1/4 to 1/3 of the population across Central Asia and northern India, and falls to almost nothing among indigenous populations of the Americas. Some indigenous groups in South America are essentially 100% type O, with no A and no B at all. The Basque Country holds the highest concentration of Rhesus-negative blood in the world. In East Asia, Rhesus-negative barely exists. Random noise does not draw maps like that. Something has been pushing on human blood, in different directions, in different places, for a very long time. To understand what, you have to know what a blood type physically is, and the answer is far smaller and far weirder than most people expect. Every red blood cell in your body is coated in sugar chains, anchored into its outer membrane. You have somewhere around 25 trillion red cells circulating at any moment, and each one carries roughly 1 million copies of the molecule that determines your ABO type. These chains are short. The base structure, which everyone has, is called the H antigen. If that is all you have, your blood is type O. The gene that modifies it sits on the long arm of chromosome 9 and codes for an enzyme called a glycosyltransferase, which has one job. It grabs a specific sugar and attaches it to the end of the H antigen. The A version of the gene builds an enzyme that attaches a sugar called N-acetylgalactosamine. The B version differs from it by a handful of letters in the genetic code, and the enzyme it builds picks up a different sugar, galactose, and attaches that instead. Inherit one of each and both enzymes work in parallel, and you are AB. Type O is the strange one, because O is not a third instruction. It is the A gene with a single letter deleted. The most common O version worldwide is missing one guanine at position 261, which throws the entire reading frame out of alignment and produces an enzyme that cannot fold and cannot function. Your red cells go out into the world wearing the plain, unmodified base structure. That is the whole of it. The difference that killed Antoine Marouai in 1667 and killed roughly half of James Blundell's patients, and still requires every hospital on Earth to run a cross-match before a single bag of blood is hung on a stand, comes down to whether one enzyme adds one sugar to the end of one chain. Nothing about that arrangement suggests it should be preserved. Broken genes usually disappear. This one has not, and the reason it has not is the first real clue. In 2012, a team led by Laura Segurel, working with Molly Przeworski at the University of Chicago, published an analysis in the Proceedings of the National Academy of Sciences that changed how anyone could think about this. They sequenced the ABO region across a wide range of primates. What they found was that the A and B versions of the gene do not sort by species, they sort by type. The human B gene is more closely related to the gibbon B gene than it is to the human A gene. The same is true in reverse. These are not similar mutations that happen to arise separately in different apes and monkeys. They are the same ancient lineages, carried forward in parallel, through speciation after speciation, for more than 20 million years. Geneticists call this a trans-species polymorphism, and it is rare enough that only a handful of confirmed examples exist in the entire primate genome, most of them in immune genes. Consider what that requires. 20 million years ago, there was no genus Homo, no Australopithecus, no chimpanzee, no gorilla. There was an ape ancestor in an African forest whose descendants would eventually split into every living lineage of the great apes and the lesser apes. That animal already carried both versions of this gene. Every branching afterward carried them forward. When the human and chimpanzee lines separated somewhere around 7 million years ago, both walked away with A and B in the population. Chimpanzees today are mostly A. Gorillas are mostly B. Humans have both, plus a broken copy. A neutral genetic difference does not survive 20 million years. Random drift kills variation. One version wanders up in frequency, the other wanders down, and eventually one is lost, and that process usually finishes in a few 100,000 years at most. For two versions to persist across dozens of speciation events, something has to actively protect both of them. Geneticists call that balancing selection, and it means that being rare is an advantage, over and over, forever. Which raises the obvious problem. What has been chasing us for 20 million years, closely enough that the shape of a sugar on the surface of a red blood cell has been worth defending since before our ancestors came out of the trees? The broken gene sharpens the question. If type O were a single unlucky mutation that happened once in some ancient population and spread, that would be an accident of history, and there would be nothing to explain. That is not what happened. When researchers sequenced O chromosomes from populations around the world, they found that the A gene has been independently destroyed at least 3 separate times in the human lineage alone, by different mutations, in different places, producing what are now known as distinct O lineages. Each one arose separately. Each one survived. Evolution does not repeatedly break the same gene and then repeatedly preserve the broken copies, unless the broken version is doing something useful. Somewhere in the human past, and probably in many separate somewheres, losing the ability to put a sugar on your red cells kept you alive. The clearest example of that logic sits in a different blood group system, and it comes with an experiment that could not be run today. In 1976, Louis Miller and his colleagues at the National Institutes of Health published a study in the New England Journal of Medicine that answered a question doctors had been circling for decades. Plasmodium vivax, one of the parasites that causes malaria, is common across South Asia, Southeast Asia, and Latin America, and it is nearly absent from West and Central Africa, in a region where malaria of other kinds is everywhere. The gap in the map made no sense. Miller's team exposed volunteers to mosquitoes carrying vivax and tracked who became infected. Every volunteer who carried a specific blood group phenotype, called Duffy negative, remained uninfected. The parasite could not get in. The Duffy antigen is a protein on the red cell surface, and vivax uses it as a docking point. Take it away and the parasite has nothing to hold. It circulates in the bloodstream, fails to invade, and dies. In 1995, Christophe Tournemire and colleagues in Paris identified the mutation responsible. It is not in the protein coding part of the gene at all. It is a single-letter change in the promoter region, in the binding site for a transcription factor called GATA1 that switches the gene off in developing red cells while leaving it working everywhere else in the body. A surgical deletion in exactly one tissue. That variant is present in more than 95% of people across large parts of sub-Saharan Africa, and it is close to absent in the rest of the world. In 2000, Martha Hamblin and Anna DiRienzo, again at the University of Chicago, examined the pattern of genetic variations surrounding the Duffy gene in African populations and found the signature that a strong recent selective sweep leaves behind. The neighboring DNA had been dragged to near uniformity along with it, the way a fast-moving current pulls everything downstream. This was not drift. This was a mutation that went from rare to nearly universal because the people who had it kept living. An entire continent lost a functioning protein on its red blood cells because a parasite had learned to use it as a doorknob. Duffy explains one blood group and one parasite. It does not explain ABO, because ABO variation is not distributed the way malaria is. But malaria did not stop at Duffy. Plasmodium falciparum is the deadliest of the human malaria parasites, and it kills through a specific mechanism. Infected red cells become sticky and clump onto uninfected ones, forming clusters called rosettes that block small blood vessels and starve tissue, including brain tissue. Cerebral malaria is what that looks like in a child. In 2007, a team led by Alexandra Rowe at the University of Edinburgh, working with samples from Mali, published findings in the Proceedings of the National Academy of Sciences showing that the A and B sugars on red cells make rosetting substantially easier, and that the plain O surface makes it harder. Children with type O blood had roughly a 66% reduction in the odds of developing severe malaria compared with children carrying A, B, or AB. They still got infected. The parasite still entered their cells. What changed was whether the infection escalated into the form that kills. 66% is not a small edge. In a region where Falciparum malaria was the leading cause of childhood death, and where a large fraction of children who died never reached their 5th birthday, a 2/3 reduction in the risk of the lethal form of the disease is the difference between a lineage that continues and one that ends in a single generation. That is enough force to break the same gene 3 separate times and keep the pieces. If you're hooked on this story, make sure to subscribe and turn on notifications so you never miss an upload. Your support means everything and helps me bring you more emotional stories like this one. So the answer would appear to be settled. Type O is protective, malaria did the work, and the broken gene is a shield. Except that when researchers went looking at other diseases, the shield turned out to point in more than one direction. The Ganges Delta in Bangladesh has among the lowest frequencies of type O anywhere on the planet, and some of the highest frequencies of type B. It is also the historical home of cholera. Vibrio cholerae has circulated in the brackish water of that delta for as long as there are records of it, and every one of the great cholera pandemics of the 19th century began there. In 1985, Roger Glass and colleagues published a study in the American Journal of Epidemiology based on patient records from the Field Research Station at MATLAB in rural Bangladesh, where health workers had been tracking every illness and death in a defined population for years. Among people infected with cholera, those with type O were considerably more likely to develop the severe, dehydrating, rapidly fatal form of the disease. Later work has supported the association repeatedly, including studies of household contacts of cholera patients, where infection rates were similar across blood groups but severity was not. Cholera can kill a healthy adult in under 12 hours through fluid loss alone. In a delta where the disease returned in seasonal waves for centuries, a blood group that made the difference between a survivable episode and a lethal one was under exactly the same kind of pressure that malaria applied in West Africa, pushing in the opposite direction. There is no best blood type. There is only which pathogen has been in your ancestors' water and air, and the map of that history was first drawn, of all places, in a military camp in the middle of a world war. In 1918 and 1919, Ludwig and Hanna Hirschfeld were working as physicians with the Allied armies on the Macedonian front near Salonika, in what is now Greece. The camp held soldiers and laborers from across the world, brought together by the machinery of the war. The Hirschfelds recognized what was in front of them and started taking blood. They tested roughly 8,000 people from 16 national and ethnic populations, and in 1919, they published the results in The Lancet. The proportions were not the same everywhere. Type A ran high among the English and the French. Type B ran high among Indians and among soldiers from Southeast Asia. The Hirschfelds had produced the first population map of human blood from a war zone using a hospital tent and a supply of serum. They also produced something they did not intend. Their paper included a ratio of A to B they called a biochemical index, and within a decade, race theorists across Europe had seized on it as a laboratory measurement that could sort populations into hierarchies. It failed at that completely, because the variation does not respect any of the boundaries such theories require. The frequencies shade continuously across geography. Every population contains every type, and two neighboring villages can differ more than two distant continents. What the map actually records is a mixture of two things: disease, in the places where a specific pathogen pressed hard enough to leave a mark, as with Duffy in Africa, and chance, in the places where small groups of people carried a limited sample of the available variation into a new landscape and multiplied. The Americas are the clearest case of the second. The founding populations that crossed from Siberia through Beringia were small, and whatever mix of A and B those particular families carried was the only mix their descendants had to work with. Across thousands of years of isolation, most of that variation was lost. Indigenous populations of Central and South America run overwhelmingly to type O, in some groups without a single A or B individual on record. Farther north, some plains populations, including the Blackfoot, show unusually high frequencies of A. Same continent, same migration, opposite result, driven by which handful of people were in which founding group. That leaves a question the ABO gene alone cannot answer. If these sugars sit on red blood cells, and red cells stay inside blood vessels, how would a stomach bacterium or an intestinal virus ever encounter them at all? The answer is that for most people, the sugars are not confined to blood. A separate gene called FUT2 controls whether the same A, B, and H structures get built onto the surfaces of your mucous membranes and secreted into your saliva, tears, gut lining, and respiratory tract. About 80% of people of European descent carry a working copy and are what serologists call secretors. Roughly 20% carry two broken copies and are non-secretors, meaning their blood type exists in their blood and essentially nowhere else. In 2003, Lisa Lindesmith and colleagues in Ralph Barrick's laboratory at the University of North Carolina published a study in Nature Medicine in which volunteers were deliberately exposed to Norwalk virus, the original strain of what is now called norovirus. Every non-secretor volunteer was completely resistant. The virus attaches to those secreted sugars to establish infection in the gut, and in people who do not produce them, there is nothing to attach to. Norovirus is one of the most contagious pathogens known, capable of infecting a person from a dose of a few dozen particles, and 1 in 5 Europeans is functionally immune to entire strains of it because of a gene that has nothing to do with immunity. The same surfaces show up in the stomach. In 1993, Thomas Boren and colleagues published work in Science demonstrating that Helicobacter pylori, the bacterium responsible for most peptic ulcers and a substantial share of stomach cancer, uses an adhesin protein to grip a blood group antigen on the cells lining the stomach. The strength of that grip varies by which sugars a person builds, which is part of why duodenal ulcer rates have been consistently higher in people with type O for as long as anyone has been counting. So your blood type is not only in your blood. In 4 out of 5 people it lines the gut, the airway, and the mouth, which are precisely the doors through which infectious disease enters the body. The polymorphism sits at the entrance. The hardest version of this story involves a different blood group system entirely, and its stakes were not measured in years of life, but in whether a pregnancy could produce a living child. In 1940, Landsteiner, by then in New York, and his colleague Alexander Wiener described a new antigen using antibodies raised against the blood of Rhesus macaques. The Rhesus system turned out to be enormous, with dozens of antigens, but one of them, D, mattered more than the rest. Roughly 15% of people of European ancestry lack it entirely, because both copies of their RHD gene have been deleted. The consequences appear in pregnancy. If a woman without the D antigen carries a fetus that has it, inherited from the father, then blood cells crossing the placenta during birth can expose her immune system to a protein it has never encountered. She makes antibodies. The first pregnancy is typically unaffected, because the antibodies arrive too late. The next one is not. Those antibodies cross the placenta and attack the fetal red cells directly, and the result, hemolytic disease of the newborn, ran from severe jaundice and brain damage through stillbirth. Before the introduction of preventive anti-D injections in 1968, it killed thousands of infants a year in the United States and Britain alone. Now push that mechanism into the deep past. In 2021, Sylvana Condemi and Stéphane Mazières at Aix-Marseille University published an analysis in PLOS I of the blood group genetics of 4 archaic individuals whose genomes had been sequenced to high quality: 3 Neanderthals, from Vindija Cave in Croatia, from Chagirskaya Cave and the Altai region of Siberia, and 1 Denisovan from Denisova Cave. Earlier work by Carles Lauieza-Fox and colleagues in 2008 had already suggested that 2 Neanderthals from El Cidrón in northern Spain carried the O allele. What Condemi and Mazières found in the Rhesus system was that these archaic individuals carried RHD variants that are rare or effectively unknown in living human populations. Their proposal follows directly. When Neanderthals and modern humans met and had children, which the DNA of nearly everyone alive outside Africa confirms they did, some of those pregnancies would have paired a mother of one lineage with a fetus carrying an antigen from the other. Hemolytic disease of the newborn would have followed, generation after generation, in exactly the pregnancies that mattered most for the survival of a small and thinly spread population. Nobody involved would have understood any of it. A woman who had already borne one healthy child would lose the next, and the next, for reasons invisible to everyone present. Multiply that across the thousands of years in which 2 human species overlapped in Europe and Western Asia, in populations small enough that the Chagirskaya genome points to a community of a few dozen, and it becomes one plausible reason the archaic contribution to modern genomes sits at a few percent rather than half. The Neanderthals did not vanish because of blood incompatibility. Climate, competition, low population density, and simple absorption into larger groups all had roles. But among the mechanisms working quietly against them, a mismatch between the surface proteins of red blood cells may have thinned every hybrid generation, one pregnancy at a time. Given all of this, the modern claim that blood type governs which foods you should eat can be measured against evidence rather than argued about. Peter D'Adamo's book proposing blood type diets appeared in 1996 and sold in the millions. In 2014, Jingzhou Wang and colleagues in Ahmed El Sohemi's laboratory at the University of Toronto tested it using dietary and cardiometabolic data from 1,455 adults genotyped for ABO. Some of the diets did improve health markers. They improved them equally regardless of the blood type of the person eating them, the opposite of the prediction. The Japanese tradition connecting blood group to personality, which traces to a 1927 paper by Takeji Furukawa, has fared no better under testing. Where ABO does show up in modern medicine, the effects are real and modest. In 2020, a genome-wide study led by researchers in Kiel and published in the New England Journal of Medicine identified the ABO region as one of 2 locations in the genome associated with respiratory failure in severe COVID-19, with Group A carrying somewhat higher risk and Group O somewhat lower. Small effects, detectable only across thousands of patients, and consistent with everything else about this system. The advantage of any given blood type is never large in a single life. It becomes decisive only when a pathogen sweeps through a population, over and over, for 1,000 generations. Step back far enough, and the ABO system stops looking unusual, because it is one of more than 40 recognized human blood group systems carrying over 300 distinct antigens. Most of them are proteins with ordinary jobs that were never meant to be identity markers. The Duffy antigen is a receptor that mops up chemical signals in the bloodstream. The Diego antigen sits on band 3, the protein that shuttles bicarbonate and chloride across the red cell membrane and lets you carry carbon dioxide back to your lungs. The Kel antigen is an enzyme. Each was named only because someone somewhere had an immune reaction to blood that was not their own. And every one of them varies between populations for its own tangled reason. Some of that variation is vanishingly rare. In 1952, Y.M. Bende and colleagues in Bombay described patients who lacked the H antigen entirely, so their cells had no base structure for A or B to build on. The Bombay phenotype tests as O on a standard panel and will destroy transfused O blood on contact. People who have it can receive blood only from others who have it, which is why rare donor registries exist, and why one phone call can start a courier flying blood across an ocean. The single most striking thing in all of this is the age. The mutation that switched off the Duffy protein in Africa is recent, on the order of tens of thousands of years. The deletions that produce Rhesus-negative blood are recent too. But the split between A and B was already old when the first ape that could be called an ancestor of ours came down out of a tree. It predates walking upright, stone tools, fire, language, and every human species that has ever existed, including the ones that are gone. It has been carried through every extinction and every bottleneck our lineage has passed through, because in every generation, in every environment, whichever version was rarer had a small advantage against whatever was currently trying to consume us. Your blood type is a record of that siege. It is not a trait you have so much as a position your ancestors held, kept in play, because a species that all wore the same surface would have been a species a single pathogen could finish. The variation was the defense. Landsteiner found it in 1900 by mixing serum on a glass plate, and it took another century to work out that the thing he had found was not a medical convenience, but the oldest surviving argument between our ancestors and the organisms that fed on them, still unresolved, still being conducted on the surface of every cell in your body. Thank you so much for watching until the end. If you loved this story, check out the other videos on your screen now. I think you'll really enjoy them. And don't forget to subscribe if you haven't already. See you in the next story.